Alright, so welcome back to another deep dive into the fascinating processes of molecular biology. Today we're summarizing and expanding on the critical steps of gene structure, transcription, translation, and the regulation of gene expression. This is not just an overview. By the end, you'll have a detailed understanding of how our cells orchestrate these molecular symphonies. So let's get started with the foundation. Gene structure. What is gene structure? Let's break down the structure of a gene, the fundamental unit of heredity. At the 5' end, we have the promoter region. This is where transcription factors and RNA polymerase assemble to kickstart transcription. The start site follows, marking where RNA synthesis begins. Then there's the open reading frame, which stretches from the start codon to a stop codon. This region directly encodes the protein. Surrounding the ORF are untranslated regions, UTRs, at both ends. The 5 UTR regulates translation initiation, while the 3 UTR can influence mRNA stability and localization. Finally, transcription ends at the termination signal, signaling RNA polymerase to disengage. The process of gene expression involves two major steps: transcription and translation. Transcription converts the DNA sequence into a complementary RNA transcript. This step occurs in the nucleus in eukaryotes, but in prokaryotes it happens in the cytoplasm. Translation then reads the mRNA transcript to assemble amino acids into a protein. This happens in the cytoplasm at ribosomes. But before we dive into these steps, let's discuss the essential similarities and differences between transcription, replication, and translation. Replication, transcription, and translation share many similarities, but each serves a distinct purpose. Replication duplicates the entire genome. This is essential before cell division. It begins at specific sites called origins of replication, ORI, and uses enzymes like DNA polymerase to synthesize a complementary strand. Transcription focuses on individual genes, creating an RNA copy. It begins at promoters and uses RNA polymerase to synthesize RNA in the 5'3" direction. Translation converts the mRNA sequence into a protein. Ribosomes initiate translation at the start codon, AUG, and synthesize polypeptides from the N-terminus to the C-terminus. What's remarkable is the precision of these processes, ensuring the faithful transmission and expression of genetic information. Transcription is the first step of gene expression, and it occurs in 3 distinct stages. Initiation: RNA polymerase binds to the promoter region with the help of transcription factors. In eukaryotes, this includes the Tata box, a conserved DNA sequence upstream of the start site. Elongation: RNA polymerase moves along the template strand, synthesizing a complementary RNA strand by adding nucleotides in the 5'3" direction. Termination: in eukaryotes, transcription often stops at a polyadenylation signal, AAU-A, which leads to the release of the nascent RNA transcript. In prokaryotes, termination can be row-dependent, involving a protein that disrupts the RNA-DNA hybrid, or RO-independent, using a hairpin loop structure in the RNA. Eukaryotic RNA requires significant processing before it can leave the nucleus. First, a 5' cap is added to protect the RNA from degradation and facilitate translation. Next, a poly A tail is added to the 3'N, further stabilizing the molecule. Finally, splicing removes non-coding introns, leaving only the coding exons. This step is mediated by the spliceosome, a complex of small nuclear RNAs and proteins. Once mature mRNA reaches the cytoplasm, translation begins. Initiation: the small ribosomal subunit binds to the mRNA, guided by the Shine-Dalgarno sequence in prokaryotes or the Kozak sequence in eukaryotes. The first tRNA, carrying methionine, binds to the start codon. Carrying methionine binds to the start codon. Elongation: the ribosome moves along the mRNA, and tRNAs deliver amino acids to the growing chain. Peptide bonds form between amino acids, powered by the ribozyme activity of the ribosome's large subunit. Termination: when a stop codon enters the ribosome's A site, a release factor binds, prompting the release of the completed polypeptide. Gene expression is tightly regulated at every level. At the DNA level, methylation silences genes by blocking transcription factors or recruiting chromatin remodeling complexes. During transcription, promoters, enhancers, and transcription factors determine which genes are transcribed. At the RNA level, RNA interference, RNAi, can degrade mRNA or block its translation. Regulation ensures that cells only produce the proteins they need, saving energy and maintaining homeostasis. Even after translation, proteins often undergo post-translational modifications to become fully functional. These include phosphorylation, which can activate or deactivate enzymes, glycosylation, which aids in protein folding and cell signaling, ubiquitination, which tags proteins for degradation. Additionally, chaperone proteins ensure proper folding, and some proteins require cleavage to become active, as seen with insulin. Let's recap the key points. Replication ensures genetic continuity by duplicating the genome. Transcription converts specific genes into RNA transcripts. Translation reads mRNA to synthesize proteins. Regulation occurs at every step, from chromatin remodeling to post-translational modifications. Test your knowledge. Which process involves removing introns and joining exons? A. Replication. B. Splicing. C. Translation. D. Transcription. Think it through. The answer is B. Splicing. This essential step ensures that only the coding regions of eukaryotic RNA are translated into proteins. Take your molecular biology knowledge to the next level with King of the Curve. Visit kotc.me for interactive quizzes, detailed lessons, and expert insights tailored to ace your exams. Your success starts here.